On behalf of NyBerMan Bioinformatics, it is my great pleasure to extend a very warm welcome to each and every one of you to today's webinar, where we have focused on the leverage of machine learning for the prediction of drug targets. In today's session, we will embark on an in-depth exploration of machine learning. Our journey will encompass the stages of conducting data analysis, constructing a machine learning model, testing it, and ultimately applying it for the prediction of drug targets in cardiovascular diseases. So before moving ahead, please allow me to give a brief introduction to our NyBerMan Bioinformatics. Established in 2019, NyBerMan Bioinformatics has earned a global reputation as a leading bioinformatics service provider with a distinctive and expansive vision. Our primary mission is to create an inclusive network that connects academia, industry, students, and active researchers worldwide in the realms of bioinformatics, biology, and related fields. This vision became a reality with the launch of NyBerMan Bioinformatics Europe in 2021, broadening the reach of our exceptional training and services to a global audience. Our expertise spans a diverse array of disciplines encompassing omics, structural bioinformatics, computational drug design, molecular modeling, molecular dynamic simulation, cheminformatics, programming, artificial intelligence and machine learning, systems biology, health informatics, and intellectual property rights. Throughout the years, we have orchestrated a multitude of impactful workshops and skill development programs across various bioinformatics domains, complemented by an extensive portfolio of 200 plus webinars, each attracting an average of 300 plus participants from India and beyond. Our commitment lies in fostering collaborations and offering opportunities for project outsourcing, consultations, freelancing, and training activities. For further information and to get in touch, we welcome you to explore our website www.nyberman.com. Now, I'd like to give a brief introduction to our series, the ReproducibiliTea Hands-on Lecture Series that convenes bi-monthly with a special emphasis on guiding participants through practical sessions aimed at reproducing established bioinformatic pipelines. This inclusive platform extends a warm invitation to individuals at all academic levels, whether pursuing bachelor's or master's or Ph.D. degrees, or conducting a research in the field of bioinformatics and its related disciplines. So if you are enthusiastic about staying informed or would like to be the part of our series, we encourage you to subscribe and learn more by visiting our website at www.nyberman.com/webinars. So before moving ahead, there are few steps that you guys have to follow so that we can ensure that only registered participants receive the certificates. So the first step is to request the feedback link. In order to do that, send us a direct message on our Instagram page requesting the certification feedback link. And please do not send it multiple times if you have already sent it once. Second is the feedback link distribution. The feedback link will be provided during the webinar, that is between 6:30 p.m. to 7:30 p.m. IST. The third step is the response time for Instagram message requests. So we will accommodate Instagram message requests until 11:50 p.m. IST on Saturday, 11th of November, 2023. And our fourth step is the duration of feedback link activations. So this feedback link will be active from 7 p.m. to 12 a.m. IST. So dear audience, please meet our guest today, Dr. Nirupama Singh, a successful microbiology student turned expert in structural bioinformatics and data science, holding a Ph.D. in biotechnology and bioinformatics from University of Delhi. With 6 years of hands-on research and development experience, Dr. Nirupama stands out in these dynamic fields. Her journey is marked by a robust foundation in machine learning and Python with 5 years of expertise. Proficient in Linux and cloud servers like AWS, she excels in structural biology, systems biology, protein and gene network analysis, data mining, and computational genomics. In pivotal roles at Netaji Subhash Institute of Technology, she developed machine learning models for predicting drug targets in cardiovascular diseases. Nirupama Singh is a trailblazer, an unwavering researcher, and a visionary in bioinformatics and data science, an invaluable asset to our data-driven world. We consider ourselves extremely grateful and fortunate to have Dr. Nirupama with us today for this webinar. And one important thing, dear audience, please ensure that you record or write all your questions that comes to your mind. We'll address all of them in a Q&A session later. So you can post your questions in the chat box later. So Dr. Nirupama, I'd now request you to please take the session ahead.
Okay, so yeah, hello everyone. Thank you for joining this session. So today I'll be showing you how you can actually make use of machine learning to predict the biological drug targets. So let me just start with the introduction. I'll give you a brief introduction about drug discovery, processes, challenges, and role of machine learning, that how machine learning comes in the picture. So drug discovery is basically a complex and time-consuming process involving the identification of potential therapeutic targets, screening compounds, and it involves an extensive testing before the actual market launch. Okay. And what is the problem with the traditional drug discovery? That is that it involves high cost, lengthy timelines, and low success rate due to a trial and error approach. This is where the machine learning comes in the picture. So machine learning is something that has revolutionized this landscape by analyzing large biological and chemical data sets, identifying intricate patterns, improving the efficiency in predicting drug targets as well as drug target interactions. So it helps in accelerating the overall drug discovery process by giving it a structured pre-processing, or you can say before actually doing the experimental work, you can get a structured pipeline through which you can enhance the accuracy of the drug targets you will identify. It can streamline the lead optimization by enabling virtual screening, and it also reduces the need for extensive experimental iterations that are being used in the traditional drug discovery. So it ultimately provides you a more efficient and a structured and cost-effective pathway for the development of new pharmaceuticals. Yeah. So what is the importance of drug targets? Why do we have to identify drug targets? So first of all, I'll tell you that what are drug targets. They are the biological entities. They can be either protein, enzymes, or any receptor that are there in the human body. And these are the drug targets should be those that play a pivotal role in various cellular processes and pathways in the human body. And what is the significance of these drug targets in drug discovery is that they have the direct influence on the efficacy and safety of drug compounds that are being used to target them. So by modulating the activity of specific proteins or receptors, the drugs can exert precise effects on biological processes associated with a particular disease. So this is this is a brief introduction about drug discovery and the importance of drug targets. Why do we identify the drug targets and all of that? Now, what are if you actually want to do the drug target prediction with the help of machine learning techniques, then what are the steps going to be? So the first step would be data collection. So the data you will collect could be it has to be the biological data, and it could be chemical structures, known drug target interactions, or some protein data. So any kind of biological data you can use. Now, this next step is the feature extraction. So suppose if you're working on protein data, then you have to extract the relevant features of those proteins that will represent the overall data for the machine learning algorithms that you are going to use. So you have to gather the you have to make a one learning instance in such a way that you have a particular data point as well as the feature related to those that data point. Then the third step is going to be the model training. So once you have the data in the form of learning instances, so you can actually make machine learning models by training the ML algorithms on that data, which will act as your training set, and you will make some ML algorithms, ML models. On the basis, you can use several machine learning algorithms such as random forest, support vector machine, logistic regression. So on the basis of your purpose, like whether you want to regression or whether you want to do classification, you can choose the machine learning algorithm and you can perform. You can train the algorithm on your data. Now the fourth step is validation and testing. So once you have the trained model, you have to validate the efficacy of that model first. If you find that the efficacy is good enough, then you can proceed further with the testing of that model. Like you can give the model the unknown data set, which is not seen by the model, which is unseen for the uh, model you have prepared, and you can actually test it on the unknown data set for either predicting a value or a prediction of a class. So for example, if you are making a classification model, you can classify between two categories. So the model I'm going to show you today is a classification model itself. Yeah, so this is the case study I am going to take. So this case study talks about use of machine learning for prediction of drug targets for microbial CVDs. So microbial CVDs are those cardiovascular diseases that are associated or influenced by microorganisms. So these are the particular class of CVDs that are like known that these are important, but they are not well studied yet. So I have taken this case study, and this is the first block of this case study. So in this first block, what we have done is we have taken the protein data, like the proteins that are involved in microbial CVDs, and we have extracted the features for those proteins. So in this case study, I have taken physicochemical features of those proteins, which are already there in the numerical format. So if you want to take other features as well, such as sequence features, functional features, then you have to convert them. You will get them in a textual format, and you can convert them in the numeric format by using several like you can do that in Excel as well. You can make use of Python libraries to convert them into the numeric format, because model gets trained on the machine learning models that you are going to make will identify the numeric data. It will not work on the textual data. So I extracted the features from UniProt and ProtParam, and the initial processing was done in Microsoft Excel. Now this is the second block of the whole pipeline. So the next step was to make the ML classification model. So as I've already told you that I have made a classification model for predicting drug targets. So what I'll do is I'll make a model which will be able to classify between drug targets and non-drug targets. And I will do it in the Python programming, making use of these Python libraries such as Pandas, NumPy, Scikit-learn, and Matplotlib for visualization of plots and several other things. So yeah, this is the Python environment. I'm going to show you that how to actually make a machine learning model in this Python environment. So this is a Spider software. It gives you a Python environment, and it will be using Python 3.8 version. And I have already made a script. So now I will open the script and will walk you guys through the each and every step of the machine learning model we are going to prepare. So this is the Spider environment. Here you can write your script and save it for further use. Here you can see the variables that are being created while running the script. And this is the actual console where the script is being run and the variables are being created. So first of all, what you have to do, you have to define the, select the library where your files are present. Then you can open them. Yeah, so this is the script. So first step is I'll import all the libraries and modules that we are going to use. So these are the, so what is a library? Library is a, like, so for example, scikit-learn is a library, but pre-processing and model selection are its modules. So it is like this. Now, first of all, as the first heading, first comment says, so whichsoever line is that starts with the hash is a comment. So the first comment is that import libraries and modules. So in this step, I'll import the libraries and modules that are being used in the creation of the machine learning model. So as you can see that all the modules and the libraries have been imported. Now the next step is to load the file, that the training file that you will use to make your model. Yeah. So the training set file or the data set has been uploaded, and you can also have a look on it from here only. Like these are the protein IDs and these are the features. And the last column is the target label. Like when you train a machine learning model, and this is specifically a case of supervised machine learning. So in the case of supervised machine learning, you have the features as well as the targets. So the last column is the target, that you are training a model on a data set which already knows that which one is the drug target and which one is not a drug target. So yes and no signifies that this first protein is a drug target and the third protein is not a drug target. So this is about the data set, the training data set, which we'll use to make the model. Now in this step, I have uploaded the file, and we can actually see that file from here. Yeah. So if you can see that first column is this column shows the protein IDs, and these are the features. And the last column is the target label. So yeah, I have already told you that what is target label and what are the features. So yes and no signify for the drug target and non-drug target respectively. Yeah. Now in the next step, we'll separate the target labels and the features. Yeah. Now if you can see, two more variables have been created. So in the feature variable, you can only see the features. There is no entry column or the target column. So these are the only 14 features that are there in the data set. And if you see the target column, then you can see the only one column, which is of target. Now what we will do is, so if you can see the target is in the textual form. So now I will do the label encoding. So the next step is label encoding, and we'll convert this yes or no into 0 and 1. Yeah. So if you can take a look on target underscore bin, then you can see that yes and no have been converted to 0 and 1. So that model can read what is the target label. Now, next step is feature scaling. So why do we do feature scaling? Because let me just show you once. Yeah. So if you can see that here the range of number is very low, and if you see in the third column the range of number is very high, and in this instability index column the range of number is 0 and 1 only. So to bring them in a single range, we have to do feature scaling. So now I'll run the set of feature scaling. And if you open the scaled features, then you can see that they have come in a single range. Now the next step is to split this training set into train as well and the test set. So when you make a machine learning model, you have to split the whole of the learning instances learning instance points into training and test set, in which the model has already seen the training set points, but it has not seen the test set points. So it will give some other accuracy on the test set. Like you are testing, first of all, you are training on the actual training points, and then you are testing it on the smaller subset, which is the test data set. So I am splitting the overall data set into 70 and 30%. So 70% is going to be the training set and 30% is going to be the test set. Yeah. So four more variables have been created. So the X test and the X train, this X train is the splitting of features. Like you can see here that in the training set, 2380 features have gone and 1020 features have gone in the test set. And the Y train, Y test are the splitting of target labels. So it has to be the same as the training set. Now what I'll do is I'll make a random forest model, machine learning model on this splitted training set. So you can make like I am just showing it to you for an example, but you can make several different models on this data set, such as logistic regression model, support vector machine model, k nearest neighbor model. But for just an example, I'm showing you that how to make a random forest model. And also these n estimators, maximum depth of the model. And to identify the values of these, you have to run one additional step, which is hyperparameter tuning, but it takes a little longer time. That is why I haven't kept that step in this script. But there is a module, grid search CV, which you can use and perform the hyperparameter tuning, and you can identify these parameter values. So I've already run that and I have identified the values. That is why I'm proceeding with these values and will make the model. So I guess the previous steps I have run are clear. Okay, so in this, I have made the random forest model. Now we'll predict the accuracy of the model. So as I said that we have to validate the efficacy of the model. So we use several accuracy measures. So first one I have used is accuracy score, and the second is the ROC AUC score. Yeah. So now you can see we have got the 99% accuracy on the training set and the 72% accuracy on the test set. So why is the accuracy high on the training set? Because these are the instances which have already been seen by the model, and the test set instances have not been earlier seen by the model. So that is why accuracy is low on test set. But this doesn't mean that accuracy cannot be increased on test set. You can definitely increase the accuracy on the test set by increasing, adding several more features, as well as performing cross validation, and you can take into account so many other things to increase the accuracy of the model anytime. Now, like we'll proceed with this accuracy only. And now I will, once we have made the model, we have identified the accuracy on two accuracy measures. Now, what I'll do is I'll identify the important features, like whatever features I have used for training this model. I'll identify that which feature is contributing most in the prediction. So I'll make a plot and I'll take a look at that what features are important in the identification. So let me just save this plot and show it to you. Okay. So as you all can see that this particular library has I have now identified all the important features and least important features that are there in the model. So the instability index value is contributing most in prediction accuracy of the model, that it has the most important role to play, and the least valuable role is of estimated half-life. So this feature is of least importance, and the instability index value feature is of the major importance in the identification of the, like, in the accuracy of the model. Yeah. So we have made a model. We have identified its accuracy. We have identified the important features. Now what we'll do is, next step is that we'll run it on the prediction data set, like, which is not seen by the model. Which so this prediction data set are the proteins which are involved in microbial CVDs, and our ultimate aim is to identify drug targets for these CVDs. So we have the protein data and their features for the microbial CVDs, and now we'll use our model we have prepared to identify the drug targets that we can target for uh, to overcome these microbial CVDs. Yeah. So in this step, I'll upload the prediction data set. Then I'll define the features. Then I'll scale the features, because the training set features as well as the prediction set features has to be the same in order to so that model can work on the prediction data set, because the data has to be the same. Now in this step, I have uploaded the prediction data set. I have scaled the features of the prediction data set, and I have uh, used the model to predict the drug target on the prediction data set. So if you can see this C named file, then this one says that this protein is a drug target, and 0 says that this protein is not a drug target. So let me just show you the file I uploaded. So the file name was df2. Yeah. So this is the file, like this is the prediction dataset file. And as you can see that this is very much similar, not very much, this is similar to the training set file. So then only the machine learning model would be able to work on this file. And I've also scaled the features of this file as well. And the predictions have been made on these proteins, and you can see the predicted target in this file. Now I'll extract this file and show it to you. So first of all, I'll extract the predicted targets file. And one other thing I'll do is that I'll identify the confidence score of the prediction. Like if the model is saying that this is a drug target, what is the confidence? Like what is the confidence that this is actually going to be a drug target? So in this step, I'll identify the probability and confidence score of the predictions that model is making. So I have run that step as well, and I have now extracted two files. One is in which the model has identified the targets, and another one is in which the model has identified the confidence score of the predictions. So now let me just show you both the files. Yeah. So this prediction.csv. Yes, I have some other. No, sorry. Yeah, now I did not run that step. I have run it now. Okay. So let me just show you all the three files. So this is the prediction set file in which there are proteins as well as the features of the. So these are those proteins that are involved in microbial CVDs in causing CVD effects, and we have to identify drug targets out of them. Now I'll what and this is the file in which model has predicted that which one is the drug target and which one is a non-drug target. So I'll just paste the first column in that file. Yeah. So now you can see that which protein is a drug target and which one is not. And third I talked about is the probability of the predictions. So here you can see that 0 is for non-drug target and 1 is for drug target. So let me just paste that file here, column here as well. Yeah. So like this is the main file which we are going to use, because if the model is saying that this particular protein is a drug target, then what is the confidence of that prediction? So as you can see, that model is quite a little dicey about this prediction, that it is somewhat showing that 55% is the confidence score. The model can say that this is a drug target. So we are not going to consider this as a drug target because model is dicey about this. So we'll apply the filtering and we'll only take the high confidence score of the predictions. Yeah. So as you can see, the model can say with the 100% probability that this is a quite good drug target because the confidence score is very high. So what we can do, we can just filter till the 0.7 because 0.7 is considered as a high score. So all those proteins that are having confidence score above 0.7 can be considered as the most probable predicted drug targets. So yeah, this is all about the predictions, predicting the drug targets using, like I have told you that how to import the modules. Then you have to upload the training set. Then you scale the features and you can do trains test splitting. Then you can make the model. You have to identify the accuracy of the model. You can also identify the features. Then you can actually make the predictions on your prediction data set. Yeah. So this was it about the making, like this is how you can predict the drug targets for any disease. Like you can collect the proteins and the feature associated with them, and then you can make use of machine learning models to predict the drug target for any disease. Yeah. Now I would like to take the questions from the audience if anyone has any sort of question regarding this.
Okay. So, yeah, we extend heartfelt gratitude to Dr. Nirupama for her amazing presentation. It was remarkably engaging and undoubtedly left a lasting impact on our audience. So we would warmly invite our audience to share their questions and queries pertaining to the discussed topic in the comment section. We will be more than happy to address them post the discussion of the few pre-collected questions. So, Dr. Nirupama, there were a few questions that we had earlier. So the very basic question from the discussed topic, I think, would be, how do we discover these drug molecules? Can we discover them in silico, or how do we do that?
The drug molecules or the drug targets?
No, the drug molecules.
So the drug molecules can be chemically synthesized, and there are several databases from where we can get these lead compounds or the drug molecules. So the already known drug molecules can also be used, as well as their analogs can be used, and also their analogs can be synthesized. And yeah, so if you talk about the drug molecules, you can take it from databases, or you can chemically synthesize and then can prepare a library for these drug molecules.
Okay, so as you said that the methods we can through which we can know these drug molecules, through this we must have thousands and thousands of molecules for a particular maybe target, or in order to target a particular disease. And so is
Right.
there a way to screen them through AI in a very short period of time? Because as you said initially that in order to screen a lot of molecules, it will take a lot of time. So how AI
Yeah,
can
right.
be helpful in screening these thousands and thousands of drug molecules?
Okay. Okay. So what is the traditional method that earlier we used to do the virtual screening of all these drug molecules against a particular target? Now we can prepare, like AI has given us opportunity that we can make machine learning models which can be trained on already known drug target interacting pairs, and then they can be used for predicting the new drug target pairs. Okay, so we
Okay.
don't have to perform the virtual screening of all the molecules against a one target. We can give the model multiple proteins and multiple drug target pairs, and we can simply identify that this drug is best suited for which target.
Okay. Okay. And since now we know how powerful these AI ML tools are, so a lot of them had this question in their mind that being a student, how can they master in these skills, and how can they use it as a bioinformatics student? This was one of the most popular questions I saw.
Okay, yeah, this must be a popular question. So, if you are a student, then you can just start looking in the literature that what people have already done, and you can just have a prototype of your work. Like, if you are reading a research paper, then you can have an idea that okay, these all the steps they have taken into consideration, and I can also extract the features from this database or some tool. Then if once you have the data, the first and foremost step is to collect the data. Once you have the data, then there is no limit. You can like what you can apply to that data. So the main step is to extract the data. So to extract the data, you can make use of several bioinformatic databases, as well as research papers are also there. Some research group, they also provide supplementary data with their research findings. So one can make, if student is reading a research paper, he or she can look for like whether supplementary data is being provided. So he or she can just take the supplementary data and can further use it for making the machine learning models and to carry out the further studies.
Okay. Okay. So do you think that in future maybe we can have AI designed drug doses? Like AI is telling you the best drug and the best target for it, and it is also giving you the doses, appropriate doses. Is that ever
Of
possible?
course,
What do
like
you think?
yeah, it is not just going to happen in the future. It is being used nowadays as well. Like there are certain models
Okay.
which can tell you that this is a drug compound which you can definitely use for this particular protein, and not for a particular protein, but if you have heard about drug repurposing, that a single drug can be used to target multiple target proteins. So it is not just that one drug can be used for one protein only. It can be
Yeah,
used, it
absolutely.
can be repurposed, and it can be used for multiple proteins. And if you talk about the dosage concentration, like in what amount this drug should be given, if you're talking about this, then that could also be used in training the model. And of course, it will be able to tell that what the drug is going to target which protein, and in what concentration would be required to hinder the process of that protein.
Okay. Okay. Another most famous question that I saw was how students from core biotech and other life sciences background like zoology and all, can they make their future in this field? Because I think you're the best person to answer this, because you yourself are from microbiology and then you had a career here, you have built it in the field of bioinformatics. So they also wanted to know how is it possible and how to do it more efficiently.
Yes, definitely, because I am the live example. So I was from microbiology background, but when I
True.
entered in this field, so I was, yeah, so I was not having any knowledge of bioinformatics apart from this word bioinformatics. So, but I had an interest in this field. So I switched my field from wet lab to dry lab. And
Okay.
what I started is I started reading the stuff which is related to bioinformatics first. Then I entered into the programming field. So first
All
I
right.
started my journey by reading about bioinformatics and the tools and databases and techniques that are being used in bioinformatics, the traditional techniques as well as what is new in this field. And then I entered into the programming field. Then I merged both of them together. And then I read the relevant literature where programming is being used, programming, or you can say machine.
Um,
This was my journey that by reading the relevant literature, I got to know that how can machine learning be used and how can I learn machine learning and can implement for my objective. So during my PhD itself, I got to know that, okay, machine learning can be used for this purpose. And I actually implemented it on my data. I collected the data from several sources and I implemented it for the prediction of drug targets.
okay, so all in all, we can say it's all about the interest. If you have a strong interest and passion towards
Yes,
it,
if
yeah.
anyone has the interest in this field, he or she can definitely enter into this field and can do wonders.
Okay, true. Thank you so much. Thank you so much, Dr. Nirpan. I'd like to take few questions from the live chat box. So one is from Gigi Smith. So the person wants to know about the modules that you have used in your scripts, a brief introduction about it, and what's the purpose of those modules, and how do you know what is those modules used for, as in what is that particular module for?
Okay, okay.
Yeah.
So the pandas and NumPy are the very basic modules. Not these are not the modules; these are the libraries of Python. So pandas is used for used to load the data in the Python environment. So if you have the data set in the form of CSV or Excel file, you can load your data with the help of this pandas library. So it is used to load the data. Now, scikit-learn is a library which is specifically dedicated for machine learning. So it has all the machine learning algorithms, not just random forest, but as well as support vector machine, logistic regression, linear regression, decision trees. So there are several machine learning algorithms that are there in the scikit-learn library. Yeah, so this and yeah, so pandas is used to load the data. Scikit-learn is specifically used for machine learning, and the matplotlib is used for visualization. So I just showed you one plot in my code. So for that, I used matplotlib. And NumPy is used for to convert your arrays into series. So we did not have to use that module, but that module is used for that purpose.
Okay. So another question is from Atul Tiwari. He wants to know what properties of data should have, a data set should have.
Okay. So a
Yeah.
data can have a lot many properties. For example, if you If you're working on sequence data, then you can have the sequence of that protein as a data property. Okay, you
Yes.
can feed with that data itself. And like I have worked on the several features. So I included functional features, sequence features, and physicochemical features, network features. So what were network features? So before actually performing machine learning modeling on my data, I also constructed a network for these proteins for the host pathogen interactions which were there. So I
Yeah.
computed the topological parameters for the network, and I incorporated those topological parameters as the features in my data. So there is no limit that what kind of data you can use. You have to do like hit and trial methods. You have to include every possible data feature you can. And then probably you can see that which data features are performing best for your objective.
Okay. Okay. The next question we have from Arun Bahadur. He wants to know, because since you were talking about the ML model evaluation, which is one of the important steps. So, how do we evaluate our ML model when there are few negative samples present, and or maybe they are completely absent? And another question that the same guy has is, how can we solve this problem when we have ML models when they're multi-target problem targets for a drug. When there are multiple targets for a drug, then how can we solve it using machine learning models?
Okay, so let me just answer the first question first.
Yeah.
So you cannot make the model if you don't have both the positive and the negative data set. So the model I just prepared has both the kind of data sets. It also had the positive data sets, which was of drug targets, and it also had the negative data sets, which was a non-drug target.
Okay.
So you have to have both the positive and negative data sets to make an efficient machine learning model. Because what happens is, if you train a model only on the positive data set, it is not going to classify between the drug target and non-drug target, because it has only seen the data of drug target. It has not seen that how a non-drug target looks like and what are the features of non-drug targets. So we have to give the model the features of both drug targets as well as non-drug targets, or you can say the positive features as well as the negative features, so as to make the model learn that these are the kind of features of a positive instance, and these are the kind of features of the negative instance. Okay. And the
Yeah.
second question is to
So
target
do you,
multi. Yeah.
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 The question was, how do we solve the problem when there is multi-target drug? So how can machine learning help us solve that particular problem?
Multi-target. So what is the problem? I am not able to get the problem.
The question is, how can we use these machine learning models
Hmm.
to solve the multi-target drugs? Like a particular drug has a multiple target, and we want that drug to work on one single target.
Okay, on one single target.
Yeah. Yes.
Okay, so for that what we can do is we can let me just think on it.
Yeah.
Yeah, so in this case we have to make the model learn that we can provide the features for all the proteins a drug is targeting, and we have to make the model learn that if uh, suppose you want to make the drug work on only one target. Okay, so you have to make the model learn that these are the features of the target I am looking for, and only if these are the features are there, the model has to tell that okay, you have to target this particular protein only, not the rest of the proteins. So there are certain, you have, first of all, you have to identify that what is the difference in the features of all the proteins a particular drug is targeting. Once you identify those differences, you have to train the model accordingly on the basis of those difference. So the first step is to identify the difference between those proteins a particular drug is targeting. Once you identify the difference, then only you can make the model learn that this is going to be the difference. And on the basis of this difference, your model will tell that this is the target protein and this is not.
All right. All right. Got it. Okay. So the next question is from Roman. So he has this question that if protein has its own parameters, so if there are no cavities or the target site on a particular protein and the score in machine ML is 1, what should we do in such cases?
First of all, I don't think that model will tell that this is a drug target because because the features will feed to the model will also have the feature of the binding site, that this is the feature and this is the kind of binding site a drug target should have. So we have already trained the model that this is the feature of binding site. So and also, even after providing this information, if the model tells that okay, there is no binding site and your model is telling that this is a drug target, then you can first do blind docking and you can look for the possible binding sites on that protein. And also there is another thing which is targeting protein interactions. Like if that protein is not targeting with a drug molecule, it might be interacting with another protein and gaining some conformational change. So you can target the interaction in that case. First of all, it will not tell that this is going to be a target protein. And if it tells, then you can probably target the protein interaction in that protein.
Okay. Okay. The next question is again from Arun Bahadur. He's saying that how did you choose non-drug targets for microbial CVDs in the test set to build random forest model?
Okay. So to make the non-drug target set, I chose those proteins that were having the major role in the normal biological processes of the normal human body. Okay. So I chose those proteins and non-drug targets. I took the whole proteome data I removed the proteins that are already known drug targets because they are going to act as my positive data set. Then I did random sampling. And first of all, I did random sampling. And after doing a random sampling, I discarded those proteins. Those were having important role in the human body, like normal human physiological processes. So yeah, and I also performed 5 times cross validation and 5 times random sampling to make these models.
All right. Okay. Okay. Now, this is the last question. It's from Shrutika Jatla. She wants to ask that is there any AI web-based standalone tool that can predict interaction between target and a drug?
Yes, there is a database named DisGNet, which gives you the association between several biological entities such as drug, disease, drug, and the drug, protein and disease, and the enzymes and receptors. So it is a database which is named DisGNet. It can give you associations in several biological entities.
All right. All right. If I would request participant, they can unmute themselves in case they want to ask any question. They can unmute themselves and ask. 1 or 2 more we can take. I hope, Dr. Nirupama.
Yeah, that is fine.
Anybody?
Good evening, ma'am.
Good evening. What's your name?
Ma'am, myself Nikita.
Hi, Nikita. Good evening.
Ma'am, basically, I'm from MBIPC background in my intermediate, and now I'm doing my engineering degree now. and I'm actually interested in learning the things which really converges my biology background with engineering. What would you suggest me to do now?
Yeah, so if you want to pursue a career in the field of biology as well as engineering, then bioinformatics is the domain for you. Because yeah, so this is the domain which makes use of biological data and which merges the biology and informatics together. So if you want to carry forward with the biology as well as engineering, you can definitely switch to bioinformatics field. So what is the field you are currently studying in?
Ma'am, electrical and electronics engineering, ma'am.
Okay, okay. Yeah, so you can definitely, so you can start working on biological data sets, and you can learn programming languages, and then you can merge both of them and proceed with the bioengineering field.
Okay, ma'am. Thank you, ma'am.
You're welcome.
Hello, ma'am. Myself, Ovik. So I have a question. That you make a sample Excel sheet. So where the protein have the drug target. So that is for particular one drug, or that may be a different type of drugs.
That could be for different types of drugs. So I did not take into consideration the drug molecules, but they were the drug targets for a disease. So I did
Okay.
not take into consideration that which drug can be used to target them. But my study was limited to the drug targets only, not the drug molecules.
Yes, but the thing is that this information means which protein is for which drug target, that is available in Uniport, or everything is available in Uniport.
So this information is not available in UniProt, that whether this protein is a drug target or not. So if you want to know that which are the already known drug targets that are there, you can go to the drug bank database. Okay. What I have done is I use the proteins present in drug bank database as the positive set, that these are the already known drug targets. Okay. And then the proteins I had were novel, like they were not classified as drug targets or non-drug targets. So what I did was I made the classification model and classified them into drug targets and non-drug targets. From UniProt, I extracted the features for those proteins.
Okay, and this is totally manual process for features extraction.
No. So for feature, so UniProt gives you the result in the batch, in a batch. Like if you give a set of protein, it can you can retrieve all the features for that particular list. But the physicochemical features I extracted were from Prot Param. It is a tool of XPC. So it takes input of one protein at a one time only. So for that I used web scraping. So I wrote a web scraping script, and I ran that script to extract all the features in one go from the Prot Param tool.
Okay, suppose I make a sample Excel sheet for a particular means suppose in case of human, and I am working in other organism, means other aquaculture organism. So can I mean suppose that from this can I make a prediction possible? Like repurposing of drug in some aquaculture animal.
Like if a drug is working in human, you want to check whether it is working in another organism.
Yes, yes.
Yes, you can definitely do that.
Okay, so for that purpose, if I got some research paper where they already give the sample data set by making themselves, so I take this data set and then run my proteins that are associated with my aquaculture species associated disease. So then I can some result. So it is okay in the means this procedure is okay.
Like you already have the sample data set, right?
Sample, yes, sample data set from previous any study means somehow someone publish a paper and who also give the sample data set.
Yes,
So I
yes.
take this sample data set.
Yes, you can definitely take the sample data set, and you can also modify it on the as per your requirement. Like what is your objective to do with that data set? So you can anytime modify that data set according to your objectives, and you can definitely work on that data set.
So okay, after getting some heat, means some drug target, then I have to need to docking and stimulation like this thing, right?
Yes, you can do molecular docking. You can do if you want. So these are the traditional techniques. These are the traditional techniques: the molecular docking and the molecular dynamic simulation. So if you have less number of drug molecules or the proteins, you can go for molecular docking or virtual screening. And then once you identify the relevant proteins and targets, targets and drugs, then you can also do the molecular dynamic simulation.
Okay. So any other technique is there? Means as you say, this traditional technique.
Yes, so these are the traditional techniques. So you can also think of making some machine learning models which can identify where you need not do the virtual screening. You can just simply train the model on already known drug target interaction pairs, and then you can just give the data of these unknown interactors of drugs and target proteins, and then you can predict the interactions between them.
Okay. Okay.
Yeah,
Thank you, ma'am.
so
Thank
you
you
have
very
to
much.
make a model which can predict the drug target interaction pair.
Okay. Thank you very much.
Good evening, ma'am.
Good evening.
Ma'am, my question is, you are again and again telling the known drug targets and prediction data. So, is it comes under the supervised training of the machine learning? Because whether we like when we develop some machine learning algorithm, we need both the supervised learning and unsupervised learning. So, is it coming under the supervised learning, the known drug
Yes.
targets?
Yes. So it is not that the known drug targets comes under the supervised machine learning. So what comes under the supervised machine learning is that when you have the features as well as the known outcome. So for example, in this case, I had the features, and I already, when I prepared the training set, I had the features, and I already knew that if this is the feature, if it is a drug target or not. Okay. So when you have the target label known, then you apply supervised machine learning. And when you don't have the target label, then you apply unsupervised machine learning. Because in case of unsupervised machine learning, you don't know what is the target label, and what is the outcome for a particular instance. Okay. So for example, if I give you a protein, I tell you all the features, and you don't know further, like whether this protein is a drug target or not, then you have to apply unsupervised machine learning on that data of proteins. Okay. When you just have the proteins and the data. So you can apply unsupervised machine learning to identify patterns out of that data. Okay.
Okay, ma'am. And then we have to compare it with the supervised,
Yes.
and then we have to
Yes.
go for
So
the further
once
processes.
you apply unsupervised machine learning, you will have the understanding about the data, and then probably you can further apply supervised machine learning on it once you have the target label. So to implement supervised machine learning, you have to have the target labels.
Okay, ma'am. Thank you so much, ma'am.
Okay, you're welcome.
Okay. So now we'll end up the Q&A session. Thank you so much, Dr. Nirupama, for addressing the questions. Dear viewers, the feedback form is open now. And I have an exciting news for all of you. Soon, we are going to have a virtual internship on machine learning aided structural bioinformatics. The details will be shared on NyBerMan CAD WhatsApp group or the WhatsApp community. So please stay tuned. Please be in contact to know for further details like date and time. The input file and the handouts will be given in this virtual internship. So you can consider this as a Diwali gift for all the viewers from the NyBerMan Bioinformatics group. So as our webinar draws to a close, I would like to express our sincere appreciation to Dr. Nirupama for generously contributing her time to this event. Her insightful session is certain to have illuminated the minds of aspiring students, scientists, and researchers in our audience. We are confident that our attendees have significantly enriched their understanding of advantages of machine learning for drug target prediction through the session. So, dear
Yeah,
audience,
yes.
thank you so much, Nirupama.
And thank you so much, everyone, for joining this session.
Hope to meet you in other webinars too.
Yes, yes, sure, definitely.
So, dear audience, you will soon receive a feedback that you have already received in the chat box from our Instagram team. Please ensure that you provide your correct email ID name when completing the feedback form. Additionally, we kindly request that you share your certificates on your social media platforms, tag us, and help us spread the word about our upcoming events. Before leaving, I want to once again express my gratitude to each and every one of you and extend my best wishes and a very happy and prosperous Diwali on behalf of NyBerMan Bioinformatics. We look forward to your active participation in our future webinars and events and workshops, internships. So until next time, take care.